Hi, everyone. My name is Max Cohen. I'm from the Chapman Lab at UCSF. Thank you for the opportunity to speak with you today about work that we're doing into TGFβ-dependent expression of SFRP and other genes in fibroblasts of human lung tissue. This is the Chapman Lab. We've been fortunate to have a relatively mild course of things here in San Francisco and had a lab lunch out in Golden Gate Park recently, went for a walk. We're appreciative, of course, for generous support from the NIH, through especially the U01 PCTC. The work I'm going to tell you about is driven by a hypothesis that TGF-beta signaling in fibroblasts is important in driving expansion of fibroblasts signaling that contributes to fibrosis, dysfunction of fibroblasts and of epithelium. This work is based on a long interest in the lab in understanding TGF-beta receptor signaling, TGF-beta biology. Over the last number of years, the lab did a screen to identify inhibitors of TGF-beta signaling in EMT, and more broadly, and identified a class of compounds called trihydroxyphenol compounds that are modified by the LOXL2 enzyme to a active intermediate that can inhibit the TGF-beta receptor kinase. And this inhibition of the TGF-beta receptor kinase then blocks signaling. And LOXL2 itself is also blocked. Now, because LOXL2 is a fibroblast-specific gene, this leads to fibroblast-specific inhibition of TGF-beta receptor kinase signaling. And so, of course, this is summarizing quite a bit of work that's been done by the lab, including the publication that Yingwei had in JCI there. And the trihydroxyphenol compounds are widely found in the plant world. EGCG is one such compound that is naturally occurring and available over the counter as a supplement. This version of it, T-Vigo, is a brand that's available online. Amazon and other retailers sell. It's a 95% purity that's extracted from green tea. And so there's quite a few trihydroxyphenols, but this one in particular is just readily available. So in a proof of concept study, patients were given EGCG prior to surgical lung biopsy that they were going to get for the purpose of diagnosis for illness of ILD. So these are patients seen in the ILD clinic with unknown ILD cause. Their CT scans and their clinical histories weren't sufficient to explain why they have illness, and their treating clinicians decided to obtain a biopsy. And for this, some patients elected to participate in research where they took EGCG prior to biopsy, and others didn't, but still donated lung tissue. And so what we can see here is that for patients, and then we analyze lysates from these patients. And so control lung lysates versus surgical lung biopsies from fibrotic patients who didn't take EGCG versus did. We can see that in the untreated biopsies, there's increases in profibrotic biochemical markers, collagens, snail, and SMAD shown here. And these are decreased in patients who took EGCG prior to their biopsy. And when that's quantified, you can see that it's increased. And the red and the black dots are separating it out by IPF versus non-IPF diagnosis on the subsequent pathologist read of the result. So it affected both. So EGCG for 2 weeks prior to surgical biopsy reduces pro-fibrotic biochemical marker activity quite substantially. What we've continued to do since then was obtain these samples as we've been able to, and do single cell RNA sequencing. We make a single cell preparation of the lung tissue and perform single cell sequencing of FACS sorted cells, half epithelium and half mesenchyme. And the rate is that about half end up being IPF based on our referral patterns and et cetera. And so here, what I've done, I'll talk you through this. It's not too complicated, but it takes a moment, is I took 6 biopsies, 3 that were treated with from patients who took EGCG prior to biopsy and 3 that didn't. one donor lung that was not used for transplant as a control, one IPF explant. I took the Haberman-Kropsky publication and the Skui-Shepard publication. I took all the mesenchyme from these samples, combined them together, and then I subsetted to just the fibroblasts. And here I'm showing the total cell number for fibroblasts, and I'm showing a UMAP with the unsupervised clustering of these fibroblasts. So this is just fibroblasts. Smooth muscle parasites are already removed because we're really interested in understanding the fibroblasts, because that's where EGCG acts. You can see there's 11 clusters, and when I examine these clusters for known markers for different types of fibroblasts, I get an annotation that is shown here. And we don't need to get too far into the weeds on this part, but what I'm going to point out are that in red here, what I'm calling the CTHRC1 high, this is a collagen modifying gene that this publication identified and shows it really marks the fibroblasts at the highest expression of collagens and the most pro-fibrotic, we think. In green here, that says 1 are the alveolar fibroblast populations, and in blue here, SFRP2 is marking these populations, which are a mixture in normal lung, I think, probably of this is marking peribronchial and adventitial fibroblasts. There's 3 unbiased clusters, and we're going to go into what SFRP2 is in a minute. And then there's some other fibroblasts that are really, not as well understood and not as much the focus of this talk. Now, the first question is, in patients who took EGCG prior to their biopsy, what effect did it have? Well, if we look in the effects on TGF-beta signaling, because it's a known inhibitor of TGF-beta, or that's what we think it is doing, we wanted to understand, is it really inhibiting TGF-beta? So I'm showing here a feature map of CTHRC1, this collagen modifying gene that is a marker for profibrotic fibroblasts, but also a TGF-beta responsive gene. And in the control lung, these are donor lungs not used for transplant. There's a little bit of expression in fibroblasts from this area, but there's a marked expansion in explant. In control biopsies, so biopsies from patients who did not take EGCG, the TGF-beta inhibitor, you can see there's quite a bit of expression in the population that I annotated. And here in the EGCG exposed fibroblasts, you can see visually it's pretty clear there's less. When we do a quantification here, a visual quantification with violin plots, you can see the EGCG biopsy versus the control biopsy, there is less. And I'm going to show that the numbers on the next slide. Looking at some other TGF-beta genes, you can see decreases in collagens in SNAIL2 and MMP2. So this is quite promising for an effect on TGF-beta signaling in fibroblasts. These quantifications are just comparing all cells from this condition and all cells from this condition. And now here, this is the differential expression of all cells from the EGCG fibroblasts versus all fibroblasts from the biopsy control sample. And so you can see TGF-beta 1 and 3, snails, the collagen triple helix related containing 1, a collagen modifying gene, collagens, and other TGF-beta responsive genes that went down by substantial margins by expression level, by percentage of cells that express them in the EGCG versus control biopsy, and the adjusted p-value for multiple comparisons for these genes. So this is pretty impressive for an effect of downregulation of TGF-beta by EGCG, which is what we expect based on the prior research that identified EGCG as a TGF-beta inhibitor. Now, when I took this differential gene expression list, a much longer list than just these genes, and put it into the IPA analysis algorithm, IPA identified a number of upstream regulators that would have predicted these changes. And it says that indeed TGF-beta is inhibited to explain these changes, but it also identified P53, MYC, and beta-catenin. So it's predicting downregulation of all of these. So this is, again, quite promising that we are in fact downregulating TGF-beta in fibroblasts. And when we look farther, well, what else is changing? In addition to TGF-beta, we can see that there's impressive effects on quite a few Wnt genes. As I said before, the beta-catenin is predicted to be downregulated by IPA. When we look at specific Wnt genes in the differential expression analysis, comparing the fibroblasts from the EGCG exposed condition versus the control biopsies, we can see quite a few Wnt pathway genes that come up. And so that's what I'm showing here. You can see in particular SFRP2 has the strongest effect. SFRP2 is a secreted fizzled protein, and so it contains a frizzled domain, and SFRPs are modulators of Wnt signaling. I'll get into this in a little more, but so this is a modulator of Wnt signaling, and it's markedly downregulated by EGCG. And if we look at it visually, it's quite clear comparing the control biopsy versus the EGCG biopsy that there's marked downregulation. And again, other Wnt-related genes have substantial changes here. Looking at the expression on the UMAP profile of SFRP2, we can see that SFRP2 in control donor lungs is primarily expressed in this cluster, which is what I annotated as the SFRP2 positive cluster here. But in explants, it's actually quite widely expressed. So not only in those cells, but in a much wider range of fibroblasts. And it's also seen in the control biopsy. But in biopsy from patients that took EGCG, there's visually one can see that there's this downregulation of SFRP2 back, much more like the control lung, the donor lung, than like the control lung. So it's a pretty impressive effect on SFRP2, which goes with the differential expression quantification that I showed on the last slide. Now, SFRP2 itself is, there have been a number of publications and it's not exactly clear what it does. So we wanted to start by determining if it really does affect the Wnt pathway and if it's an activator or an inhibitor. SFRP2s have generally been, or SFRPs, excuse me, have generally been thought to be inhibitors of the Wnt signaling pathway. And so we use the top flash reporter. This is a promoter with a luciferase driven by a Wnt responsive promoter, and transfected it into the 293 cells and looked at the effect of SFRP2 on it. And you can see that when Wnt3 stimulated cells upregulate the luciferase reporter in response to a signal, and when SFRP2 is added at different doses, it will increase that signal. It doesn't downregulate it like an inhibitor, but it increases it. And so that's shown here with the upregulation of luciferase activity at 3 different doses. Now, what's quite interesting is that on its own, it doesn't seem to have an effect. And so if we just look, for example, at the 60 nanograms per mil concentration, that on its own, as opposed to the unstimulated condition, no difference clearly. SFRP2 on its own doesn't stimulate Wnt pathway reporter changes, but when it's combined with Wnt signaling with a frizzled agonist, it has a significant effect on increasing it. Which brings me back to the finding that we see EGCG biopsy versus control biopsy, that comparing the full differential expression profile in IPA, there's a predicted effect on the Wnt pathway. TGF-beta pathway and Wnt pathway both come up. And so in summary, what I've shown today is data that patients who have pulmonary fibrosis and consume EGCG, a trihydroxyphenolic compound, prior to biopsy, they have robust downregulation of TGF-beta responsive genes in their fibroblasts. And we've identified SFRP2 as a TGF-beta responsive gene, and that it is a potentiator or increasing Wnt signaling. And this suggests that TGF-beta is promoting Wnt pathway activity in fibroblasts, at least in part through SFRP2. Now, we're continuing to study this and particularly interested in how it is driving dysfunction of progenitor cell populations, as others from our lab, Dr. Lasaw and Dr. Kathiria, are discussing in their talks. Thank you, everyone.